talk today hopefully will give some ideas about how to optimize your current specimen management or biorepository operations, or if this is something you don't have, to get some ideas around implementation, as well as what kind of vendors may be out there to provide those needs for you. So today's talk, why end-to-end management for biological specimen management, how we do things at Genentech, and then lastly, key considerations for implementation. So for why the end-to-end? So here what we're really needing is samples from patient to analytical result, and there are a couple factors that we're looking for to try and ensure that we're having great specimen management. The first is to reduce variables in the analytical process. So we get these samples in, they're going to go out to diverse labs, maybe some internal, some external, even though it might be one protocol different groups are using. The variability from platforms, the variability from assays, from who's actually running that platform. You're trying as much as possible to get samples to not be another big variable that you're adding to the process. The next idea is that effective specimen management done in an end-to-end manner will enable novel science through integrated logistics. And the third idea is trying to solve a so-called attention gap related to specimen management. And I'll go into that a little bit more later. So in specimen analysis, again, there are tons of variables, and hopefully the sample management variables aren't contributing to the variability you're seeing in your analytical results when you're trying to look for patterns in your data sets, when you're trying to make go, no-go decisions on new drugs, on biomarkers. So having solid practices all the way through the process about, are you going to standardize collection, are you going to standardize handling, how can you standardize how you're storing and delivering to labs, this helps the end results to be more usable during the research period. So enabling novel science. Historical methods have been less demanding of sample management groups. If you're looking for using ELISAs in plasma or serum to look for a certain biomarker, if you're just doing genotyping of DNA, the timelines aren't as tight because often these will need to go into a clinic situation. These biomarkers are often more robust, but a lot of your novel I'm going to say analytical tests, especially in the earlier research phase, are not going to be as robust. If you're looking to do genotyping analysis on some circulating tumor cells from whole blood, you may only have a few cells in that entire 2 ml vial, and this is being shipped halfway around the world. If you do not have good processes to enable the scientists to say, yes, we found the cells, yes, we're able to do genotyping, if you don't think about how it's collected, how it's transported, chances are these new scientific techniques won't be working at your company. Then I like to call this the attention gap. So people generally don't think about any of the variables related to sample management until the data analysis phase or until they're trying to make a decision for go, no go. And this is why it's really great to have a specimen management group at your company or to have a really dedicated specimen management vendor who you can rely on, because they're thinking about the end-to-end from the beginning and not just hoping everything went right at the end. And I will say, even we have a specimen management group, we have multiple in the Roche group, there are still fire drills, but if you leave up all these to the individual researchers to figure out, if you leave this up to your analytical labs to provide all the guidance, you're going to have a lot more fire drills. So for Genentech, we have an anchor workflow that does map to the majority of our physical and informational needs. I would say it's probably in the 60%. If you were looking at the 80-20 rule, it's not quite that, but it is a lot of our samples. This is really important that your anchor process be really refined, an efficient workflow, and cover all the parts of sample management. And this means both a physical component, but also the information component. As John was alluding to, if you don't have really a well-annotated samples, their value decreases dramatically. For some shops, this may only be a plurality of your work, but again, if you're having too many different workflows for different sample types, it's going to be really hard to effectively train personnel and to have, I guess, well-honed, efficient processes. But then to supplement this anchor workflow, we have divergent workflows, which usually are a single piece of the sample management puzzle. Maybe it's just the timing with handling, or maybe it's the way we're cleaning the sample information for a PK vendor that allow you to meet some, I'm going to say, divergent need without changing your entire sample handling process. So for Genentech, our anchor workflow is simply plasma and serum samples that we batch for our analytical labs so that they can kind of get them on their instrumentation and running as quickly as possible. We do this within 72 hours of receipt. We make sure that these are going to be compatible with their automated platforms so that they're not spending their time transferring tubes, identifying samples, or rearranging the tubes in a box. For us, this is usually PK, PD, or immunogenicity samples, and we need to make sure that this data can be combinable with the clinical patient data and the sample data at least 30 days prior to data reporting. So the processes that we use to meet this anchor workflow, again, standardized tubes, standardized containers, the boxes we have, making sure we have monitored freezer units for sample storage, and then for the data, really making sure that there are keys to link the data that exists in all these different databases. So we have information in our sample database. There's going to be information in assay databases. Of course, the patient data is going to be huge. You need to make sure that this can come together in the end and that you're planning for this. And lastly, methods for efficiently translating paper collection records into digital files that can be loaded into our sample LIMS. So for Genentech, we don't want to be spending the time doing tons of data entry from all of these paper requisition forms, which often are much easier for sites to maintain than in, say, an ECRF, but we do want to still have an efficient workflow. So we've devised a way where data is kind of created at a third-party site, and then we are able to efficiently import that data, associate with samples, and pass those samples along. So some of the requirements that will drive these divergent workflows for physical collection, so the core idea here is the collection of a biological sample such that it provides valuable information throughout its subsequent testing. So some of your key considerations, people were talking different types of collection tubes you might use depending on the assay. Of course, that's important. But I especially feel that as you're going into all these different sites in all these different countries, complexity of method is huge. Most sites understand how to do venipuncture and to collect whole blood, turn it into plasma or serum. If you're looking, though, for, say, a novel tissue biopsy, or if you're looking for other matrices which sites aren't really familiar with, really consider that when you're determining which sites you're looking at placing the trials. And for the ethical nature, I mean, a lot of times I feel like we still live in the world where we needed a mil per assay, so we're still collecting 12 vials of blood when we could probably get away with 3. We're still doing a lot of things, not necessarily thinking about the more that we can get kind of patients on the side of the trials, maybe we can help sway some of these ideas about what residual use can be done, how can we use these samples to really understand these diseases and as much as understand the drugs we're hoping to develop. For physical handling, the core idea, preservation of a biological sample, including both the stability and chain of custody. For here, very straightforward, key considerations, storage conditions, access control. And I'll just touch on access control real quick. This is thinking about who's touching the sample all along the way. I mean, yes, if it's packaged up, you probably don't need to be having the FedEx guy get GLP training. But if your lab is fully open for anyone to use, and especially if at a convenience sake, you're like, well, we'll have the researchers come and pick them up themselves, just consider that that could change how stable your samples are, because these are generally not personnel who are thinking about maintaining the overall stability of all the samples. And lastly, the labeling method idea. So someone mentioned sites having problems applying labels. I think we probably all spend way too much time dealing with sample labels, but on that note, it's amazing how often that goes wrong. So thinking about, are there simple ways to deal with things, color coding between forms and tubes? Are there ways to make this easier? And then finally, for the physical side, it's the disposition, giving this to your assay lab. And the key idea here is distribution of the sample such that it can efficiently provide the valuable data. So I'll really hit on that keyword efficiency. It's great that you've got the sample out your door, but if then the next group is spending all this time either entering that data by hand because they didn't get an electronic file from you, or rearranging the samples because they need to test it by concentration of the sample, then you're not really adding the value to your organization that you could be. So just thinking about what happens with a sample after it leaves your door, you're probably going to do a better job of supporting samples for your organization. To talk a little bit about the Asia side, so for shippers and couriers, if you are shipping domestically in the United States, yeah, FedEx, UPS, they may be sufficient. But if you're going to be shipping to Asia, Europe, most other countries, you're probably considering couriers who do the doorstop-to-doorstop service, deal with the customs issues, deal with the icing issues, because the last thing you want to do is, yeah, spend $1,000 procuring samples or spend hundreds of thousands of dollars procuring samples in a trial only to not spend enough money on the shipment and the samples to sit on a curbside and become useless. So I've talked a lot about the physical side, but I will say the information side is just as important. So for collection, the core idea here, collecting information regarding the specimen and its source so that it will be meaningful for later analysis. So you're looking at what systems are you using to get this information, and I would say one of the key ideas that often becomes a problem is when you're collecting all this data about a patient or about a sample, which fields are free-form fields that you just want the information, and which ones do you need to have controlled so that when you mash all this data up later, you can actually analyze it in a meaningful way. If you're capturing indication or visit in an open manner, then actually trying to plot PK over time or looking at with treatment does a patient's biomarker go down, it's much harder if you're allowing too much flexibility in the data you're collecting about the samples. For information handling, it's managing the information from a specimen and the source, including cleaning and association. So you're going to get dirty data. The last presentation was talking about the 99% of samples that the NCI was procuring where those samples were not useful. I'd imagine some of it was a physical issue, but I'd imagine a lot of times it's that the information you have is wrong or not usable, and that's just as good of a reason to have to not test the sample or throw it out as it thawed sitting on a bench. So having a process to clean those samples so that you can turn a sample that, say, looks like it has nonsense information into a sample that then you can generate a meaningful data point for. On the controlled vocabulary note, if you deal with a lot of different departments or if you deal across institutions, across companies, controlled vocabulary is really tough. There are standards out there, but I don't see this problem getting too much better. So just to kind of recap some of the key thoughts, end-to-end processes deal with all aspects of biological sample management. And so this, again, is both the physical collection, handling, and disposition, but really just as importantly, the information collection, handling, and disposition. The goal of end-to-end processes is to reduce your analytical variables, to enable novel science, and to solve the attention gap. And then lastly, if you are going to implement this at your own site, you need to consider, is this a core competency you want to develop? Can you do so in really an org-wide approach? And what is your organization's needs with regards to flexibility and scalability?